within it is a combination of living and non-living things. We're going to be working with the biosphere. And when we zoom in, we're going to be looking at specifically an ecosystem that's going to contain the community. Now, we'll talk about the community as the next section, which includes the non-living surroundings. And for you guys that are starting to furiously write notes, it's okay. This stuff isn't super important. It's really just a high-level overview once we start to zoom in. You'll know when we start getting to that point. But right now, again, just kind of an overview. So if you can just pay attention and just relax on the pen, save it for later. When we look at the ecosystem, like for instance, when we look at the rainforest, that includes the non-living things. So the community specifically is looking at the population of animals, or not necessarily just animals, but looking at the bacteria, looking at all the simple cellular organisms, simple organisms to the complex organisms that live within. So these are the living species, or living organisms within a community. Once we look at a population, we're looking at one specific species. This is a group of organisms of one type that live within that ecosystem. So if we're looking at, for instance, that bison within that prairie, that's specifically the population we're looking at. Okay? All of that stuff is included in life sciences. But what we're going to be doing, especially in physiology, is to look at the organism and all the makeup of that organism. So we're going to be able to dissect. When we look at the organism, that organism is a compilation of all the things that are made together to sustain that organism, to make it live, whether they compromises living, eating, reproducing, to perpetuate its species. We're going to move down to the groups of cells that make the organism. So when we look at the organism as one living thing, we're going to be able to see the cell is the smallest functional unit of life, and how that cell works together to contribute to the whole of that organism. We're going to take those cells, we're going to group them together. We have tissues, we have organs and organ systems, which I'll talk about in just a little bit, that work together to organize that organ system to contribute to the functioning of that organism. Once we look at the cells, we'll be able to see that cell. We'll go over the main cell types, of which we'll be able to look at our neurons. We'll be able to look at our connective tissue. We'll look at all those 4 types of cells that work together to contribute to that organ. And then lastly, we'll look at the molecules contained within those cells, because each of those molecules, things that we have to look at, whether they're a neurotransmitter, whether they're hormones, you name it. The function, the structure of the molecule is going to be able to determine its function. And these are going to be important. So as we go through, when we look in physiology, we're going to be able to hone in on these last 3 portions right here, which we'll be able to see on the next slide. So the question that we always start off with, when we start off physiology, I already gave us a preface earlier, that physiology is not like anatomy. So the key difference here, and this is something that is important, is anatomy, you learned about the structures. You learned about the attachment points. You learned about the nerves. You learned about all the different tracks that that you needed to be aware of. Okay? You learned about the structure of the heart, the different chambers of the heart. So that is anatomy. It's the study of the structure. You understand the muscle attachment points. You learn about why the brachioradialis attaches at this point and this point. In physiology, we want to learn more about the function of it. Okay? You know the orientation of the heart. You know the chambers of the heart. What we want to learn is the function of it. How do we generate pressures? How do we get blood to eject from the left ventricle? How do we measure that? What is that implication of that measurement? How does that apply to our clinical measurements? Okay, so if you're running an ultrasound on somebody and you want to determine cardiac function, and you want to look at the ejection fraction. What does the ejection fraction mean? How does that tie in to the muscle fibers? How does that tie into the cells? Is there a disruption in any of the cellular mechanisms? Is it anything to do with the tissue? Anything to do with the organ? Anything to do with a cell? Or is it the lack of a single molecule? And you'll look at different, especially when you get to pathophysiology, it could be a dysfunction at any of these levels. So it's prudent as we go into those organ systems to talk about everything from a molecular level to a cellular level, to a tissue level, down to the organ level. And then even the organ system in order to talk about the function of the organism. So anatomy is the study of structure, Physiology is the study of function. That's a big thing on here. You see that physiology contributes to all of this right here. We talk all about everything from the molecules to the cells to the tissue, organ systems, organisms, and it even includes populations of the species. We don't go so much into population, but that stuff is really interesting if you ever want to get into it. So the level of organization, as you see, as we mentioned earlier, we will spend a small chunk looking at the molecules. We talk specifically about it at a chemical level. We'll talk a little bit about the atoms, and then we talk about how those atoms come together to create these molecules. So these molecules are going to be important because these are the macromolecules that are important to life. When we talk about DNA, we talk about all the other proteins, carbohydrates, fats. We have to understand their structure, and we'll start to draw on those as we get into those later lectures. When we take those molecules, we put them together in a cell, and inside that cell, we're gonna spend a good chunk going into the cellular structure, whether it's the functions of the organelles inside the cell. We'll talk about the cells themselves, and we'll differentiate those cells. And what do I mean by differentiation? We're talking about the specialization of the cell. We take your average cell that you learned in biology, and we'll start to talk about how they work in muscles, how they work as neurons, connective tissue, and contribute to the next level, which is the tissue level. So now we take those differentiated cells. We take, let's say, a muscle cell, and we group them together. And all of those cells need to work together. We're going to learn about the mechanisms of how those cells talk to each other, so that we can get all of those muscle cells to contract together. To give you an example, would be looking at the stomach. How do we get the stomach to start churning? You've just eaten something. You ate something about 5 minutes ago, and now it's in your stomach, and your stomach is starting to churn together. How do we get each of those individual cells to communicate with all one another in that tissue, to work together in an organ, specifically in the stomach, to start the process of digestion? Okay. So then once we look at the stomach, we'll start to take a look at the entire organ system. And when we get into our digestive lecture, which is our second to last lecture, we start from the mouth all the way out to the anus, and we talk about all the different functions that happen within. And you'll learn specifically every step along the way. And then when we talk about the digestive system, you'll learn how we work together as an organism. And as we approach towards the end of the course, we start to piece those together. And we draw on other organ systems to understand that process. So when we start to bring up some topics like what happens with high blood sugar, how does that relate to things that happen in the cardiovascular system? How does that relate to things that happen in the digestive system? So with that, we're going to just review. We're going to talk about things that happen on atom level, chemical level, cellular level, and so forth. So starting with the atom level, we'll be able to see the essential elements, again, some stuff that we'll talk about later on. And from here, don't stress too much about these, but like I said, we'll go into this in later detail. But one of the things that we want to see are the essential elements. So if you were like an outside alien and looking in and finding out on a molecular level, or really on an atom level, which atoms we use, you'll see that about 98% of all of these 98% of all of the elements that we use are these 6 elements right here, pichons. This is the first mnemonic. Might not necessarily be the easiest to remember, but for some reason you might remember pichons. It's 98% of total body chemistry. That means if I were to take a small chunk of you, chop it all up, and find out what those elements are, you'll see that 98% of that will be all these. Later on, we'll talk a little bit about the other elements that are not considered essential, but we do need minute amounts of them. To give you an idea, iodine is one of them. Magnesium, all the other elements, some of the trace elements that we'll talk about that are not the highest percentage, but still required. We'll go into the different bonds that hold these atoms together. So we take that carbon and we want to attach it to a hydrogen. So we're going to go into the different types of bonds that will hold that together. And again, we'll talk about the three main ones: ionic bond, covalent bond, and the hydrogen bond. And that's going to start that connection where we hold those elements together, we hold those atoms together, and we start to create both inorganic molecules and organic molecules. Of which the inorganic molecules we can see are gases, vitamins, and minerals. And we go into organic molecules. Again, we'll go into more details later on, so stay tuned. Organic molecules, we'll talk about these complex molecules that are created from those elements that we talked about earlier. We're going to be able to connect a bunch of these together and create organic molecules, of which we have four main ones. Glucose, our carbohydrates. Fatty acids, our fats. Our amino acids, our proteins. And then our DNA, our nucleotides. So each of these right here you're going to see are the smaller components, and then we can form bigger components from these smaller components. So these right now are really the building blocks that we're going to be putting together before we get into the cell. We're going to understand the basic structure of the cell itself. We'll talk about the different organelles and highlight the pathway where we can start creating proteins. How do cells create those molecules that we talked about? So if we take a cell, how does it start to produce extra receptors? How do we produce that plasma membrane to keep that external environment separate from the internal environment? So we take that cell and we learn about how we differentiate those. When we get into our tissue lecture, we talk about the four main types of tissues that arise from our cells. So when we look at the cell functions, now that in your head we've created the whole concept of the cell, what is a cell supposed to do? And you'll see 8 basic functions that we'll talk about as we go through our lectures. And we'll touch on each single one of these. And the whole main point of that cell is to be able to get the nutrients and the oxygen from its external environment. The cell was an interesting concept that was discovered that was able to sustain itself in even the most harshest environments of the earth, and yet was able to sustain and do all of these functions to not only multiply, and you'll see that's the last portion right there, is to be able to multiply, create more cells, and sustain itself. And the way it sustained itself is being able to extract what it needs from that external environment. Interestingly enough, that's what we do as humans. We do the same exact thing. We're able to interact with the external environment. We're able to harvest our nutrients. We're able to exchange air with the outside environment, yet still maintain our own internal environment, separate from the external. So that's essentially, when we look at the cellular function, you'll see that that corresponds to the things you're going to be able to see in the human function as well. So not only do we get the nutrients, in most cases, cells, they use glucose. We'll be able to take those nutrients and turn that glucose, along with oxygen, to transform that into energy. So in Thursday's lecture, we go into that conversion of glucose and oxygen and turn it into our byproducts. How do we harvest that 36 ATP per glucose molecule? Again, we talk about that on Thursday. Actually, no, next Thursday. How do we get rid of any excess? Things that we don't need. Things that don't really serve any nutritional benefit whatsoever. We eliminate it. We put it together. We synthesize proteins all the time. And I'm not talking about proteins like muscle protein. I'm talking about hormones. I'm talking about building receptors for those hormones. We're constantly rebuilding things. We're creating red blood cells. We're creating hemoglobin in order to be able to extract oxygen from the air. So we need to constantly produce these proteins and then exchange materials between the environments. Not only does a cell have to exchange with the external environment, we as humans have to communicate with the external environment. We need to take in oxygen and release that excess CO2 that's been burned through cellular respiration. So we need to eliminate that excess CO2 and move things in and out of the cell. The cell is going to be producing things, and we need to get that out of the cell and make its way out, whether it's making its way to the surrounding tissue or the surrounding fluid, or maybe it needs to make its way down over to the cardiovascular system. So you'll understand that whole highway. Material transport follows the same principle of moving those stuff when we exchange it with the external environment. And then respond to change. This is the big thing right here, is how do we respond to change in the surrounding environment? To give you an example of what happens when when we eat something? I know we were drawing on that example of the stomach earlier. Somewhere earlier, we're drawing on the stomach. What happens when we eat something? That's going to change the acidity in the stomach. So therefore, those cells that are responsible for producing the acids need to be able to detect that change. So if there's a decrease in the acidity, we need to bring that acidity back up. So those cells need to understand what changes are taking place and how to implement those changes, which we'll talk about in our feedback system very soon. So now that we've talked about the basic cell, we talk about differentiation, where we modify that cell. And what we'll touch upon when we look at the DNA, you know already that the DNA is the library of all the genes, all the things that were ever encoded. It's the blueprints to which we're made up. And cells will differentiate, meaning they'll look at that library and they'll cut things out they don't really need. In fact, they're not cutting it out, they're turning it off. So when we look at, for instance, muscle cells, we'll see that muscle cells are now differentiated. They're specifically geared to turn into muscle cells. There's an internal programming that happens that we don't really get into in this class. But there's a lot of interesting stuff that talks about turning on genes that specifically turn these cells into what they need to be. So what I'm talking about specifically are stem cells. If you look into where stem cells have moved since its onset many years ago, we've gone into some crazy stuff about pre-programming of the DNA to create specialized cells. We're soon to be producing nerve cells. We can produce generic red blood cells that can get injected into you. You can do tons of stuff that work specifically before differentiation takes place. That might not make too much sense right now, but when we get into our tissue lecture, that'll make much more sense later on. But the entire notion of what that specialized cell, that cell has one main function. Whether you're a muscle cell, you're to contract. If you're a nerve cell, you're meant to send an action potential, send a signal out. If you're a gland cell, you're meant to secrete something. You have one job, and that's exactly what a cell's supposed to do. Now you take those cells, you put it together, and now those cells work together. You take one muscle cell, group it with 50,000 other muscle cells. And now we have these cells that are meant to work together to contract the muscle. Okay? So we'll talk about the communication that takes place between those cells to make up that tissue, and we look at the four main types, again, which we talk about later. Epithelial, which is going to be our outside layer, our connective tissue layer that's going to hold things together, that's kind of the glue that'll hold things, these tissues together. Muscular, which is meant to contract, and then nerve cells to communicate. And again, we take it and look at the organ level. We look at specifically those four main tissues. The epithelial tissue, and you'll see on there protection, secretion, absorption. Again, more of the things we talk about later on. This is going to be the protective layer. This is the most innermost layer. You can see that when we zoom in right next to us. If this is the high acidity area, you'll see that the epithelial layer is that protective layer on that inside. The connective tissue on the second most innermost layer is going to hold it together. That holds the epithelial tissue along with the other layers, the muscle and the nerve tissue that happens on the outer layers. The muscle is going to be meant for contraction. Again, muscle made of a bunch of muscle cells all glued together. And then the nerve tissue. This is our way of communicating from higher levels of functioning, namely the brain, being able to communicate with the stomach. So this is that nerve layer that also takes place outside. And when we get into our discussions later on, we can start to learn a little bit about the dysfunction. To give you kind of a clue in right now that I'll talk about later on, the stomach. Does anybody know anything about ulcers? Well, ulcers, you'll learn about the dysfunction that happens on the epithelial layer. Well, where this tissue no longer functions to protect the internal environment from the outer tissue layers. And so we see the breakdown of this, and you see those acids coming in, breaking into the connective tissue, breaking into the muscle, and then starting to penetrate some of the nerve layers on the inside. Yes?
A random question, but why is it I've heard that when people have ulcers, they're like always constantly hungry? Is that a true thing?
constantly hungry? I have to look into that.
That's what I heard. Like I heard of people that have it, like they're never like satisfied. They'll want to eat. But, like,
That could potentially be some disruption that's happening on the nervous layer that might warrant some looking into. But look into them.
presentation mode.
There you go, see? So now that we have all these together, now we have these 4 different layers. You'll see that each of these, even though the muscle, even though that the nerve has some function to communicate, muscle to contract, connective tissue to hold together, and epithelial to protect, all these tissues come together as an organ to contribute to one major function, of which the stomach, we'll learn, is for the production of stomach acids and mechanical digestion. The key thing for the stomach, again, later we'll discuss, stomach is meant for digestion. You'll learn both the mechanical components due to the muscle, that muscle action that's taking place within the stomach, and the production of acids on the epithelial layer to contribute to the chemical function. Stomach is meant to digest. So now that you see the stomach digesting, you'll see all the other organs that come together as part of that organ system, specifically the digestive system, that all work together. So the salivary glands for the production of saliva, you'll see the esophagus for sending the food from the mouth down over to the stomach, small intestine for continued digestion and absorption, large intestine for absorption, liver, gallbladder, pancreas for the production of the other constituents that contribute to the digestion and absorption of our nutrients. So you'll see the digestive system is how we obtain nutrients from the stuff we consume and how we eliminate the rest. Okay. So what topics do we talk about? We get into the circulatory system. That's lecture exam 2. Digestive system, lecture exam, that's actually in the final. The respiratory system, circulatory and respiratory system, all part of lecture exam 3. Urinary system, part of the final, which we talk about. We don't talk about the musculoskeletal system at all. We don't really get too much into that. I'd love to go more into detail about this, but for the purposes, because we've got so much to talk about, we actually kind of eliminate that altogether. The integumentary system, skin, hair, and nails, we don't really talk too much about. The immune system is a great topic to talk about. Again, something we don't really go too much into. We talk a little bit about it in our circulatory system when we talk about blood, but the immune system, tons of good stuff on there. The rest, nervous system, the endocrine system. So nervous system, we spend a good chunk on, at least the first couple weeks on. We talk about the neurophysiology. Endocrine system is the other communication pathway that we talk about through the production of hormones. And the reproductive system, again, not something we talk too much about. So really the heart of what we'll be looking at, 1, 2, 3, 4, 5, 6 main topics as we go through. And that's really going to comprise almost everything that we're going to be talking about in this course. Then once we put those organ systems together, now we have our organism, where we have an independent being able to sustain its own function through the sum of its parts. Put it all together, we get one organism. So when we break all that down, when we look at all those cellular processes, each of these organ systems are contributing to one or more of these functions. We'll look at the metabolic functions that take place, the metabolism that takes place within the organ system. We'll take a look at responsiveness. Responsiveness to change. What things occur, change in the triangle, changes that take place in the external environment. What happens when changes occur? You'll understand the normal physiology of what happens, but what happens when something takes place? What happens to our own temperature when the room changes? Let's say the temperature drops in this room. How do we adapt to? How do we retain our body heat in order to maintain that normal homeostatic temperature of 98.6 degrees Fahrenheit? We look at movement, and I'm not talking about movement of the human body, but I'm talking about movement of cellular organisms. How do we move? How do we transport things in and out of the cell? Growth, how do we perpetuate? If we were to lose a number of red blood cells, how do we get red blood cells to grow? differentiation. How do we take our normal cells and turn that into specialized cells, and then also reproduction? So of that, now we start to get into the nitty-gritty. This is the foundation of physiology. Every physiology course that you ever go into, even I've even taken some advanced physiology classes where they say homeostasis, throw that out the window. Still talk about homeostasis. All of it, homeostasis. What exactly is homeostasis? Homeostasis is the condition of equilibrium in the body's internal environment due to constant interaction of the body's many regulatory processes. What do I mean by that? This is our balance between the internal environment and the external environment. Any changes that occur can change things in the cell, but there's always some resistance to change. Okay? So that, and I use that term loosely, resistance. I don't want to talk too much about resistance, but more so things that take place that we want to maintain normal levels of. I'll give you an example I mentioned earlier, talked about heat. What happens if the temperature drops? Our body adjusts. What happens if it gets too hot in the room? Again, our body adjusts. And there's numerous mechanisms in place that we do to maintain normal body temperature. If blood sugar levels change, how do we respond to that? If blood pressure raises, if you start to get stressed out and blood pressure starts to increase, your heart rate starts to increase. You can't sustain that for too long. You got to bring that back down. So how do you go back to a normal heart rate? How do you bring blood pressure down? If you lose a lot of blood, what does the body do to change that? How do you restore that to maintain normal functioning? You'll see that there's a narrow window that we want to maintain homeostasis. If you ever think about it, that's the reason why you ever come down to the doctor's office for something, what's the first thing they do besides doing an exam?
Do your blood pressure.
Blood pressure, vitals, yeah, they do that too. But if they don't really know what's going on, blood test. I heard blood test. That's exactly what they want to do. They want to get a blood test. Because the blood, when you think about the blood flows through every portion of the human body, flows through every cell. At least it has access to it. It has access to the internal environment surrounding the muscle tissue, surrounding the nerve tissue, everything else. The blood picks up everything on there. So the blood, when you look at blood values, they're looking for abnormal values. Where do those normal values come from? Those are the normal homeostatic values that are normal, what they expect in a normal functioning human being. And all organ systems are operating at efficiency. This is the window that they talk about. They want to look to see if your blood sugar is between 70 to 120 units. And anything outside of that may be an indication of some disruption happening. Doesn't mean a disease, it just means something is happening and warrants some further investigation, but it's where they start. So when we get into homeostasis and trying to maintain this balance, we have to look at feedback system loops. And in the feedback system loop, the entire idea is I need to sense something, right? I need to sense a change. Then something needs to receive that information and send that over to some command center and say, this is the change that's occurring. Then the command center or the control center says, if that's the change that's happening that I'm receiving from our sensors, then this is the change that's going to take place. Let me give you an example tying into earlier with our air conditioning room. There's a thermostat, right? Let's say the thermostat has the thermometer. So in this case, this could be the thermometer. The control center would be, I guess, the thermostat. Not really in, yeah, thermostat is right. And that's the computer chips on the inside of that thermostat that tells it that'll detect the changes that take place. And the effector would be either the AC unit or the heater. So that example I was using earlier is that the thermometer detects the temperature in the room. That's all the thermometer ever does. It just says, right now it's 74 degrees in here. and it sends that information over to the thermostat. Keep in mind that thermometer is not doing anything else. It's not saying it's too hot in here, bring it down. No, it's just saying 74 degrees. That's it. So that information goes over to the thermostat. And the thermostat is where you go in and you program, or maybe the thermometer if you got one of those fancier air conditioning units that will do whatever it's supposed to do, which is too fancy for me. You take those thermostats and it says the temperature we wanted the room to be is 70 degrees. Okay? So it says this is the thermostat says 70 degrees is our set point. It's going to take the information it's receiving from the receptors or the sensor, the thermometer, saying if the information the thermometer is telling me is 74 and I need it to be 70, the command center is going to say, this is the decision that I want to take place. I want to cool the room. It's going to send that, relay that information over to the next section, which is the AC or the heater. And it'll turn on the AC and say, turn this on. Okay? Now the AC is on, it's coming, it's pumping cold air into the room. That feedback, or rather the AC running, it's not going to keep running forever. Why? Because the thermometer is always reading that information. So this is where the feedback loop comes back in, where the AC, as that runs, the thermometer is going to continually detect the temperature happening in place. Now it's 72 degrees. It's relaying that information back to the thermostat saying it's 72 degrees. Thermostat says, well, it's still above the set point. Keep the AC running. And it's going to keep running until the AC keeps burning. Then we get the keeps running. Then the thermometer is going to say, now it's 70 degrees. Send that information back to the thermostat. Thermostat says 70 degrees equals the set point that I wanted it at. What do you think it's going to do at that point? If the thermostat is 70, sensor is saying 70, what is it going to do to that AC? It's going to turn it off. So you'll see how this is the entire feedback loop, and that's exactly how all of our systems take place. So looking at this, applying to what we learned, using the actual technical terms I'd like you guys to be familiar with, receptors or sensors. Receptors or sensors are the ones that receive information, or they detect the changes, or they detect the status of the external environment. And they send that information over to the control center. And that control center, you can pretty much surmise, is either a bunch of clusters of nerves, or it could be the brain itself. So it sends that information over the control center, which is going to be the brain, or nerves, or depending on spinal cord, or wherever we're looking at. And then it's going to send that information out to the effector. And the effector is, in this case, going to be the AC or the heater. So there's an afferent pathway and efferent pathway. Afferent pathway is from the receptor to the control center. The effector pathway is from the control center to the effector. And how can you remember that? This is the difference between affect and effect. And this is something that I kind of taken on, something grammatical I always see people. I actually had the pleasure, and I use that term loosely, of having a 9th grade grammar teacher who was real strict about things, and I remember her clearly because she used to knock me on not just me, but all of her students on writing things. Like, you ever see people, and I don't want to go too much on a tangent, when they write things like your, and then there, all that stuff? It really bugged her. And because of that, it really bugged my 9th grade grammar teacher. And as a result, it affects me. Because when I see this stuff pop up, I'm like, oh man, they didn't spell it right. And I get kind of hung up on that. So, looking at that, from a grammatical standpoint, affect and effect. If you want something to affect you, A-F-F-E-C-T. Okay, that's something that goes into your control center. So something that affects you goes to your control center. The effect is something that happens as a result of. And the control center is making that decision that is going to result in an effect effect. So the control center does three things. It makes a decision. It's also responsible for memory. This is probably where a mnemonic would have been helpful, because there's a third one on there, long-term something, long-term memory decision. We'll get to that third one later. So the control center makes that decision that relays over to the effector, where that effector only does one thing. In that case of an AC, it's to pump out cold air. And then the purpose of a heater sends out hot air. That's all it does. So all that works together because when that information comes in, now following what we saw here earlier, this is the stimulus. The stimulus is received by the receptor. So in this case, the stimulus would be the temperature in the room. The receptor would be the thermometer that's receiving that stimulus. The receptor sends that information to the control center. Control center makes the decision that the temperature is too high. It's going to send that output out to the effector to turn on the AC. And then what it's going to do, it's going to send that information. Once that effector turns on, the AC is on, that's going to cause a change that is going to get picked up by the receptor. So this is our feedback loop. What questions do we have on it? Yes?
What was the afferent pathway? It starts from the...
Afferent pathway is from the receptor over to the control center. That's the afferent pathway. Things that go to the control center, afferent. Things that go out of the control center is the efferent pathway. So now we look at, from that feedback loop, we want to take a look at two different pathways that we can go with the feedback loop. It's either negative or it's positive. Those are really the only two ways you can go with a feedback loop. And with a negative feedback, the whole concept of a negative feedback is to reverse a change. And to give you the best example that I talked about, I use exactly that. That was the thermostat example earlier. If it's too hot in the room, what we want to do, and if I were to kind of just tweak that example a little bit just to make this a lot more relevant to the example of a negative feedback, if we just take out the AC for all of this, right? Take out that example earlier. Thermometer says 74 or 74. Thermostat reads it at set point supposed to be 70. It's going to turn off the heater. Eventually, when it cools the room, if it gets turned to 69 degrees, the thermometer reads it, sends it over to the control center. Efferent pathway means turns on the heater to heat the room back up. So in this case, the lack of heat in a room will trigger the production of heat. Okay, that's the reverse change that's taking place. Okay, there's lots of examples that we can use in this case. Anything that involves, let me take a step back. Looking at the blood pressure, in this case, blood pressure would be the stimulus here that we're looking at. Would be received by receptors in the blood. I want to scratch that example because I tend to lose people in this example. So when we look at the pathways, let me explain what's happening in the negative feedback with reversing a change. Let me go into the example of steroid use. Example, use of exogenous testosterone use. So people that inject steroids, one of the things that we're going to learn in our endocrine lecture is the pathway for the production of testosterone, which is the hypothalamus. Again, something you don't need to know for now. Hypothalamus, pituitary, and we're going to go specifically to the testicles, which produce steroid. And in this case, testosterone is the stimulus. It's the amount of testosterone in the body. So the hypothalamus is the one that's going to be receiving the information in the brain. It's going to send it, which is also built into the control center. It's going to say, now we want to produce testosterone. So if we've got low testosterone, hypothalamus will detect it, will then produce it. Oh, I'm sorry, it won't produce testosterone, but it'll trigger the production of a hormone over to the pituitary. And that pituitary is going to send another signal over to the testicle, which is the organ that's going to produce testosterone. And what it's going to do, it's going to produce testosterone, so plus testosterone. I did not spell it, testosterone. So increase the amount of testosterone, and that comes back to the hypothalamus. So if we've got low testosterone, produce more testosterone. So what happens in the case of exogenous testosterone use or use of steroids? Injection of steroids into the system means now we've got a lot of testosterone. We don't want too much, we don't want too little. We want a narrow window of how much testosterone we use. So if I've got too much testosterone, now think about this for a second. Too much testosterone, this is going to tell the hypothalamus we've got too much. And therefore, it's going to want to do something to the testicles. Is it going to turn it on or turn it off for the production of testosterone? Turn it off. So if someone has constant high testosterone, the brain is always going to say, turn off the testicles for the production of testosterone. So people that take long-term testosterone use will find that their testicles will shrink. And I mean that. It's all testicular shrinkage is one of the symptoms of testosterone use that comes with excess testosterone production. So we want to always maintain that. And if we don't have enough of it, we produce more. If we've got too much of it, turn it off. All to keep it within that window. Now let's look at positive feedback. Because positive feedback, well, negative feedback reverses a change. Positive feedback reinforces a change. And so the example that I want to start off with is this. The number of cattle running and the overall level of panic. When you think about cows, and I don't know if you ever watched cows before. Cows are actually really interesting on their own. But if they come up and they see something really interesting, they all kind of crowd around and start staring at it. Really strange, but if you see a cow and one starts to kind of freak out, and that cow runs away, all the other cows start looking at that cow and thinking, why is that cow running? And they start panicking too. And like, I don't know why, but then that cow starts running. And then you get more cows that start running, and next thing you know, you got a whole stampede. Okay? That's a reinforcement of a change that takes place. The increased number of cattle running increases the overall level of panic, which results in more cattle running. So if you were to look at, in the case of a specific hormone, the production of a hormone starts to cause an effect, which causes more and more and more of that effect. And the best case example, which we get into later on, is birth. Is a hormone which we know is oxytocin. And so what happens in this case is the stretching of the cervix that takes place when the fetus starts to rear against the cervix. It causes stretching of the cervix. When that stretching takes place, what that control center is going to do, it's going to release oxytocin as a result of that stretching of the cervix. When oxytocin comes in, it works specifically on the uterine wall, the wall of the uterus, to cause more contractions. And those contractions presses the baby against the cervix, causing more of a stretch. And more of a stretch results in more oxytocin, which causes more of the contraction of the uterus. So you'll start to look at, if you're looking at birth, and if you were to take a mother and measure the levels of oxytocin, and as they start to approach birth, you start to see that oxytocin levels rise. And that's the example of positive feedback. Another good example is if you were to injure yourself, you get a cut. One platelet starts to stick to that area. Once platelet sticks to that area, you get more and more platelets that stick to that area to cause blood clotting. It's exactly the response that we want. So these work hand in hand. And if you were to look at both negative feedback and positive feedback, the biggest difference that happens between those two is things that happen in the efferent pathway. That's the biggest place that this takes place, where the change takes place. And looking back at that example earlier, in negative feedback, when we had high testosterone, where did that change take place? In the efferent pathway. We're turning off the testicles with the production of high testosterone. In positive feedback, oxytocin comes in and causes the contraction of the wall. Okay? And this results in an increased response. So everything else happens the same way. Receptors receive information, control center makes the decision. It's the effector pathway that's going to be different. So effector in positive feedback reinforces the change. And we all do that to maintain that window of balance that I mentioned earlier. And that any imbalance of all of that is going to result in some sort of pathology, at least for chronic issues. But if there are any temporary changes in things, we have ways of restoring that back. If your heart rate starts to raise up all of a sudden, it's got to come right back down. Okay? If O2 levels are low, we've got a way of increasing that back up. If your bladder's full and the bladder walls are stretching, we've got a way to bring it back down. Tons of ways to do it. So that should at least serve a foundation of what we're going to be talking about for the next 16 weeks together.